Assalamualaikum and very good morning to everyone. So my name is Ku Amir Ashraf bin Kumahadi. Uh, Today my group and I will talk about an important and simple method in science, which is called basic chromatography. First, what is the chromatography? Chromatography is a method we use in the lab to separate a mixture into its different parts. It works because each part of the mixture moves differently on a surface. The name chromatography means color writing in Greek. This is because the first scientist who used it, his name was Michael T.S. in 1906. Separated plant color using this method. But today, we don't only use it for colors. Chromatography is now used in many areas like chemistry, biology, food testing, solving crimes, and even checking the environment. There are many types of chromatography. For example, paper chromatography thin layer, chromatography column, chromatography gas, and also chromatography liquid. In our presentation, we will explain the basic idea of chromatography, how to do a simple experiment using paper, and where we use it in real life. We hope you will understand what chromatography is, how it works, and why it is very important. Okay, now let's continue with the explanation of the theory and also the experiment.
Hi, my name is Anik Nazahan bin Muhammad Soli. In this video, I will explain about methodology, step-by-step methodology for a simple paper chromatography experiment. Let's jump to the topic. For this experiment, we use filter paper or chromatography paper. The second, beaker or glass jar. Third, pencil. 4. Ruler. 5. Ink or food dye samples. 6. Solvent, for example water or rubbing alcohol. 7. Capillary tube or dropper. 8. Tape or paper clip. The last one, safety goggle and glove. For the methodology, we have 5 steps. The first one, prepare the filter paper. Second, apply the sample. Third, prepare the solvent system. 4, develop the chromatogram. The last one is dry and analyze. Let's go. I will explain step 1. Prepare the filter paper. Firstly, cut a strip of filter paper about 50 cm long, 2 cm wide. Next, using a pencil, draw a light horizontal line about 2 cm from the bottom of the strip. This is the baseline. Okay. The last one, mark small dots along the baseline. These are where you will place the ink or dye samples. Okay. Let's go to step 2. Step 2, apply the sample. First thing first, use a capillary tube or a fine brush to apply a small drop of ink or food dye onto the each marked spot on the baseline. Second, allow the spots to dry completely before proceeding. The last one, you can add a second drop for a better visibility. The tint is dry first. Okay, next step 3. Okay, for the step 3, prepare the solvent. Pour a small amount about 1 cm deep of the solvent. For example, solvent we use water or alcohol into the beaker. The second is the solvent level must be below the baseline of the paper so it doesn't wash the sample off directly. Okay, let's go to the step 4. For the step 4, run the chromatography. Firstly, suspend the paper strip in the beaker using a paper clip and pencil laid across the top, so the bottom of the paper touch the solvent. but the sample spots stay above it. Okay, the second, let the solvent travel up the paper by capillary action, carrying the dyes with it. When the solvent is about 1 or 2 centimeter from the top, remove the paper. Step 5. Okay, the last step, which is step 5, dry and analyze. Okay, first mark the solvent front, the highest point reached by the solvent with a pencil. The second, let the paper dry, then offset the separate color bands. Optionally, calculate the RF value for each pigment. For the formula is RF equal to distance traveled by the dye over distance traveled by the solvent. Okay, for the last part in this my part in this video is about safety consideration. The firstly, wear gloves to avoid skin contact with solvents or dye. The second, use goggle to protect your eye from the splashes. Third, work in a well-ventilated area, especially if using volatile solvent like alcohol or acetone. 4. Keep flammable solvent away from the heat or flames. The last one is dispose of chemicals properly according to your lab guidelines.
I'll be presenting about chromatography, which is a technique used to separate the individual components in a mixture. This method works based on how each substance interacts differently with two phases, which is the mobile phase and the stationary phase. The mobile phase is the solvent that moves, while the stationary phase is the surface it moves through. Because each compound has different solubility in the mobile phase and different attraction to the stationary phase, they separate as they move. Let's talk about the mobile phase first. The mobile phase can be a liquid or a gas, and it flows through the stationary phase. It carries the mixture along the stationary phase. Now, components that dissolve well in this mobile phase will move faster and travel a longer distance. That's how the separation begins. Some substances move ahead while others lag behind. Next, we have the stationary phase. This is usually a solid or a thick liquid that stays fixed in a place like paper or silica gel. Components that are more strongly attracted to this stationary phase will move slower. So again, the difference in how strongly each compound sticks to this phase help them separate as the mobile phase passes through. Finally, we measure how far each substance travels using something called the RF value or retention factor. It is calculated using this formula, which is distance moved by the solute divided by the distance moved by the solvent front. The RF value is always less than 1, and it tells us how soluble the compound is in the mobile phase. The more soluble it is, the higher the RF value. We can compare this RF value to known standard to help us identify what substances are present in the mixture.
Both samples were poured onto a strip of chromatography paper placed in a solvent. As the solvent moved upward by capillary action, it carries the soluble pigments with it at different rates depending on their solubility and affinity to the paper. The left sample produced a red spot near the baseline with a yellow streak extending slightly upward, indicating that it contains fewer components and that the yellow pigment is more soluble than the red one. In contrast, the right sample separates into several distinct colors, including blue, red, and possibly some purple, suggesting a mixture of more than one pigment. The blue component travels the highest, indicating it is the most soluble and has the least attraction to the paper, while the red remains closer to the baseline, showing it has lower solubility or a stronger interaction with the stationary phase. This chromatogram reveals that the right sample is more complex in composition compared to the left one, demonstrating the effectiveness of chromatography in analyzing and separating mixtures based on their chemical properties. Now, we can conclude that first,
Chromatography is a technique used to separate the individual components of a mixture, and second, chromatography technique were used to separate substances based on their color. The chromatography is widely used in chemistry and biology laboratories for detecting the presence of specific compounds in a sample, such as pigment, amino acids, or drugs. In the pharmaceutical industry, chromatography plays a crucial role in the purification of substance, quality control, and drug testing. It is also essential in forensic science for analyzing evidence like in toxins or blood samples. In environmental science, chromatography helps detect pollutions in water, air, and soil. Additionally, it is useful in the food industry to identify additives, contaminants, and natural ingredients. Overall, chromatography is a powerful tool for ensuring accuracy, safety, and purity in a wide range of applications. To deepen the understanding of chromatography, consider exploring other types of chromatography, such as thin layer gas and high-performance liquid chromatography, as each other offer unique method and application for separating and analyzing different kinds of samples.